students, when I go through your answer scripts for the last group work, I realized that there are some gaps in your understanding about the transcription process. So these few slides will give you an idea and help you to clear some doubts and get a clear picture. We know that in gene there are three basic components: the 5' regulatory elements, coding sequence, and 3' regulatory elements. So 5' regulatory elements, the most important one and immediately next to the coding sequence, is the promoter. What is the significance of the promoter? Promoter is where the RNA polymerase binds. RNA polymerase is actually, it has DNA binding capacity. So in the nucleoplasm, there are genes and there are polymerases, RNA polymerases, kind of floating. And whenever a polymerase encounter meets the promoter region of a gene, it can initiate transcription process. How does the RNA polymerase recognize the promoter? RNA polymerase recognize the promoter based on DNA signals. DNA signals are sequence elements. Now in a eukaryotic cell, these promoter signals, the most important ones are GC box, CART box, and the Tata box. There are some other, but these are essential. These elements are essential. So whenever polymerase enzyme see GC box followed by CART box followed by Tata box in 5 to 3 direction, it recognizes the promoter. And then it can initiate mRNA synthesis. So RNA polymerase always binds to the 5' end of the gene. RNA polymerase, once bound to the promoter, separates the two strands of the DNA double helix. And then it starts adding ribonucleotides to the 3' end. What does that mean? RNA polymerase can add nucleotides to the 3' end of the existing nucleotides. So if you look at the ribonucleotide, there is a OH group in the third carbon and a phosphate group in the 5th carbon. So how does this mRNA strand extend? New nucleotides are added to this 3' OH. So the phosphodiester bonds between the nucleotides are built between the 3rd carbon and the 5th carbon. So this strand extends in the 5 to 3 direction. New nucleotides are added to the 3'. Therefore, mRNA extends in the 5 to 3 direction. So when mRNA extends in the 5 to 3 direction, obviously the template sequence has to be read in the 3 to 5 direction because mRNA is complementary to the template DNA. So RNA polymerase read the template DNA in the 3 to 5 direction, which is complementary to the mRNA sequence. Now this strand that provides the template for the mRNA is called the template strand, which will be complementary to the mRNA sequence and which is read in the 3 to 5 direction. Now this is the interesting phenomena. If you look at the non-template strand, it is again complementary to template. Therefore, it will have the same sequence as the mRNA. Non-template strand will have the same sequence as the mRNA, but of course, thymine is replaced by uracil in mRNA. Now, this makes the life easy for molecular biologists. So, by convention, we call this non-template strand the coding strand or sense strand, because the code of the non-template strand is the same as the mRNA. So it's easy for us to study it. So whenever we give the sequence of a gene, we give the sequence of the non-template strand or the coding strand. But actually, it is the template strand that codes for the mRNA.